this way. So, I mean, I'm not going to do that work, but other people will do it. But I think what we can do, and which I think is an interesting leverage, is if we can provide the immune reagents to people, then the specialists on the anatomy of, you know, the various parts of the brain will be able to use these in their research. So that is the idea, is to really try to launch a way of generating all these reagents and then being able to distribute them to anybody who wants them. And we plan to do it. We plan to do it for the human, the macaque, and the mouse. Man, mouse, macaque. They all begin with M's. That's how we chose them. Thank you. Yeah.
But clearly, experience impinges on behavior.
Of course.
Do you think experience just changes transcription factor networks?
No. I think, you see, the connectivity will be basically established in this way, that this neuron will grow. But it will not be a wiring diagram in the same sense as it is a C. elegans. You know, there is now a great movement in Boston for a new subject called connectomics, which I think used to be called anatomy. And in fact, when I gave the first lecture a few years back in opening this program, I said they should call it anatomy, but I would understand if they change it to anatomics. But basically, I do not expect us to be able to do much with all this detailed stuff that will come out of EM reconstructions of, you know, let's say a chunk of the cortex. Because I don't think if you went to another person for exactly that region, you'd find the exact detail wiring. And in fact, I think if you went to the same person the next day, you'd find it different, all right? Because that's where the capacity of the thing is actually thought. And I don't believe that's specified. That's done by experience. Now, why I don't believe this is the following. People have two views about this. So let us ask ourselves that everything is specified. So the mathematical equivalent is there is a number somewhere in the system which is unique to that. In other words, it's a label, right? And of course, I have to specify that number. Now, you can actually specify mathematical properties by genes. For example, you build an active center of an enzyme. It's got a binding constant, and that's a number, all right? You build a sequence on the outside of a protein, and that'll give it an affinity to bind to another one. That's a number, all right? But all of that costs sequence information, which has to be evolved, and evolved by stepwise by random mutation and selection. So there is, if you like, an evolutionary cost to specify. And in fact, the argument can be turned on its head, because if there are things that vary in different cells, just because of circumstance, and there's no selection to fix them, okay, then we'll have a thing. So there's a third value. There's plus, minus, and don't care, okay? Because it doesn't mean that it is. It just is. There's no natural selection to fix that property. We don't have to create sequence information to cancel it, because it doesn't matter. That's why enkephalin is turned on in your lymphocytes when they're activated. Okay? It's not that they need enkephalin. In fact, they can't do anything with it, because the processing enzymes are not expressed in lymphocytes. And all you do is you make this clonk of denatured protein, which you sort of proteasome away, which is the cheapest way of doing it, rather than saying, well, we've got to do something for B lymphocytes to cancel this. That would be a job. So we have to beware when you make these measurements of this thing. It's good, bad, and indifferent. And I think this is very important in biology to realize that. So all we're saying is this: the kind of primitive structure which is specified by the genome, that's the one that will be done by way of this transcription code, right? And therefore, that's our job to find out what it is. Of course, there'll be a certain order with which it works. By the way, I should say that in C. elegans, where a lot of these ideas were formulated, okay, the three things are identical, namely time, space, and the code, because the decisions are taken by single cells all the time. So the three map onto each other. But in an organism with multiple cell types, they could be different. Okay? It isn't that in an organism like ourselves, that something, you cannot exclude that something which is specified first isn't at the bottom of a tree because it specifies first. Okay? Because that tree, that branch could have grown out first, and then later on the other branch. And that seems to be the case in the brain because all the interneurons are produced by secondary invasions of a primitive structure. It's true in the eye, it's true in the cerebral cortex, true everywhere. You have a structure that's there, that's generated first, and then the interneurons, if you like, are invasions into it. They usually start in a different way. So that's the thing, and then of course we must coordinate between them. So that, I think, is going to be the name of the picture. Of course, it's a hell of a program, because visualizing this and giving people tools to visualize and tools to check the evidence is going to be the main question.
Other questions for Sydney or Mark? Yes, Dan.
Give both of you a chance to talk. So how do you think about integrating what you just told us with the environment, i.e. The amount and types of food that might be consumed by the organism? How will that impact in either the complexity of the problem and or the final outcome that you're thinking about?
You want to answer that, Mark? Yes.
Mark.
Yeah.
Oh, I see. Yes. He's there. Yes. You start, because I think you can tell that.
You're talking about the neural problem. Yeah. Okay.
Is that on?
The brain. In the brain. Thinking. Well, I think that the neurons, like any other cell, are going to be dependent on maintaining a certain structure. And the differentiation of neurons is probably going to change the mitochondria, change where they're located, changes the amount of carbon that's used for construction, for generating energy, for generating neurotransmitters. All of those things are going to have an effect. So I think it's already clear that deficiencies in neurotransmitter precursors can have significant effects on brain structure. So it's only a matter of extension to suggest that anything else that can interfere with neurotransmitter metabolic pathways may also have that same effect, including mitochondrial dysfunction, including dysfunction in degradatory pathways or such. That would be the way I would answer it.
How do you have transcription factors?
Transcription factors. Well, they can be ligands for transcription factors.
Yeah.
Which may be in the nucleus, but not regulating transcription until they get the ligand.
Well, I want to just even tell you something against myself. Right, so I'm saying I just don't want anything to be contingent in this. Okay, in this first analysis,